If you want solutions, you must focus your mind. Introducing your host, Mason Hargrave.
This week, we sit down with Tom Hindmarsh Sten, a David Rockefeller Fellow here at the Rockefeller University, working in the Laboratory of Neurophysiology and Behavior under the direction of Professor Vanessa Ruta. It was originally recommended that I interview a member of the Ruta Lab by Phil Kidd in episode 3 of the podcast, and I was already familiar with Tom's work via a paper that I had read of his detailing his work on the courtship behavior of fruit flies. In it, he shows how internal state changes in the brains of fruit flies can change the way that their visual systems process information, such that they provide different behavior given the same stimulus. Say, a black dot on a screen that is meant to represent a female fruit fly for a male courter. The paper is one of my favorites that I have read over the last year, and truly is an excellent example of how experiment and theory can be combined in the context of biology. Tom has been a wonderful colleague, and I very much so thank him for coming here and joining me on the podcast. And so without further ado, I introduce to you Tom Hindmarsh Sten. So one thing that, if it seems like the theme in a lot of your work, it is the focus on having different responses to the same stimuli from the neuroscience perspective. So you're a neuroscientist, and that's kind of, if I'm understanding it correctly, one of the big questions you like to ask. Am I getting this correct?
Yes, I think that is precisely correct. I think it's such a remarkable... It's such a primitive thing about the brain that you can generate the exact same, or a different response to the exact same physical environment.
Right.
And it seems so trivial, but to me that underlies all of cognition and all of higher order neural processing.
Right.
You have to be able to do that, and without that, you could never build, a sophisticated brain.
Right. Sorry.
No, it's all good.
Mic technique here. Right.
So to me, I feel like that is it's just something that's always fascinated me. I started with as an undergrad,
right.
And I think that's the theme that I'm kind of continuing.
So because I think to most people, it's obvious why you would ask the question, given different stimuli, what different responses you get. Kind of having this map between, I do this input and I get this output from an organism. Why is it important to focus on the question of I give the same input twice and I get different outcomes?
Because I think it is one of the hallmarks of behavior, right, is that it's flexible. It has to be an animal that, if you are crossing the street, right?
Right.
And you hear a car honk, you're terrified, you jump.
Right.
But if you're sitting on a bench just by the street and you hear the exact same frequency sound, you will not jump. And that's really adaptive.
Right.
To not react and flee from something that is not dangerous.
Right.
So to be able to judge your environment depending on your behavioral context is an extremely adaptive feature of behavior. And I think that's what's allowed animals to progress so far.
Is it fair to call these... So I have a certain conceptualization of this that I've kind of gained through reading Jock Panksepp's work on affective neuroscience. And I'm wondering if this framing of things, or second F, is useful, which is that in some way that you're switching in between different affective states, and those affective states bias you towards different behavior. And to say that you have a certain bias towards, if given a certain stimulus, you have this behavior, and in a separate affective state, given the same stimulus, you have a different behavior. So is it useful to think about these as affective states? Is this something that you do? Is this what you call them?
I think it is useful in certain contexts, but it can also be muddling.
Okay.
So I think one of the most flexible of these states that we think about is attention.
Okay.
And attention is kind of decoupled from any affective, at least perceptually for humans, attention is not an affective component. It's not an affective state. But it's one of the most effective ways of, changing your response to the same stimulus. You can ignore a stimulus by not paying attention to it, and stimuli become more salient when you decide to pay attention to them.
So maybe you can break down the difference for me. What qualifies as an affective state, and why doesn't attention meet those qualifications?
I think that affective states are almost exclusively qualifiable by their kind of distinct perceptual experience that in humans.
Right.
And it is such a, anthropomorphic way to look at the world and at animals.
Right.
Because we have to impose our own experience onto them. So who knows? Maybe.
See, that's the closed loop that I'm always curious about. And I think this is where neuroscience gets to come in and being really interesting, because you're right from a behavioral standpoint. If you say, oh, that animal is depressed. How do we know it's depressed? Well, it's acting depressed. That's a circular
absolutely.
argument.
It's a completely circular argument. So I think that these affective terms are useful in terms of one of your F's, funding, right? To frame a problem in a way that it is really important for our understanding of the human brain, to understand these primitive affective states in other animals.
Right.
But in the process of trying to understand those affective states in those animals, I don't think it's a particularly useful construct to think of them that way.
Interesting. Because one thing that I have a fairly computational view of this, and I'd like to hear your comment on this, which is just the affective state can be viewed as a neurodynamical state, which is I think kind of what at the core of a lot of what you do, where you say, but you might not use the same language as I would, which is you say it breaks that closed loop. So before you say the animal is acting depressed, so it's depressed. Now with neuroscience, if you're able to actually take a look in on the inner dynamics, you could say something along the lines of the animal's acting depressed because its dynamics are behaving in this way, because it has switched into this dynamical behavior pattern inside the neurons. If you look at them, they're firing in a specific pattern, and it's that pattern that actually leads to this behavior set. And then you can switch, and if you see a sudden change in those dynamics, you say, this is a different pattern and leads to a different behavior.
Exactly. And I think that comes down to the term that we actually use, so that I think about a lot, which is internal states.
Great.
And by an internal state, I just mean, I think the best definition I've come up with for an internal state, right, is that it's a vector, essentially, pointing in different directions. Given the same input to the brain, to the early sensory systems, it's projected out into a different dimension, right?
Right.
The same stimulus can be projected along a vast array of different dimensions to yield different outputs.
So we'll take a foyer, a motion into something maybe a bit fundamental, but I think useful for the user. The word dimension gets thrown around so much in science, and it means something very specific that I think people who are more cosmically oriented start thinking it means something else, because you often work in like 10 billion dimension space or something like that, right?
Yes, absolutely. And I am not thinking about any 4D cubes or anything like that.
Right. So what do you mean as a scientist when you say dimension?
So I think dimensions are just, in neuroscience, one of the most typical ways we define dimensions, right, is that each neuron is its own dimension.
Right.
And if, say we only have 3 neurons, and those neurons can spike at 1 or 2 or 3 times a second. If I can then look at all those 3 neurons at the same time, I can say how many times are neuron 1 spiking? Then I place a dot, right, on a graph, essentially.
Yep.
How many times are neuron 2 spiking? Then I move that dot up, just like on the y-axis. And then how many times are neuron 3 spiking? And then I move that dot on that third z-axis. And that's the same way we just achieve larger and larger dimensions.
So we just take a number. If it's a value if it's a vector, as you say, that has 20 values, it's a 20-dimensional vector.
Exactly.
If you take a data point at a point in time, and it has 20 different attributes: height, weight, dimension, length, width, color, all these different things. Each one of those is a dimension. It's an attribute at a given point in time.
Exactly.
And so when you're dealing with high dimensional data, what you're just saying, right, is that there's a lot of neurons involved.
There's a lot of attributes. Exactly. There
Right.
are a lot of different parameters, if you will, that can change how this brain reacts to the outside world.
Right.
And by an internal state, we just mean that given the external world, the brain has to unfold in some way, right?
Right.
It's like a marble that rolls into the brain. And then that marble, because of the state the brain exists in, when that marble hits the marble has to follow a certain path.
Right.
And the brain can't really do anything about that.
Right.
It's only operating from, the state of the brain when the marble arrived.
Absolutely.
So an internal state then just means that what path will the marble follow this time?
Right. And it switches the pathways to end up with the marble in a different bucket, and that bucket is behavior.
Exactly.
So a stimulus as a marble comes into the machine, the marble machine at that time has positioned itself in a certain orientation, and that orientation leads to a marble dropping in a different behavioral bucket.
Exactly. And one counterintuitive thing to me when I start thinking really about this, because internal state is a word that's thrown out a lot in neuroscience.
Right.
And when you start thinking more critically about what it means to define an internal state this way, it means that memories are internal states, right?
Right.
Because memories change how the brain reacts to the environment. That is what memories are made to do.
Mm-hmm.
So I think to get into these affective states that we talk about and more of these kind of higher order temporary changes, depending on what an animal needs from the environment, or, whether an animal is horny or hungry or whatever, you need to talk about persistent but temporary internal states.
Right. And now I'm glad you said, I'm sorry, I'm just making sure. I'm glad that you said that, because one thing you focus on is horny flies.
Yes, exactly. I make my living studying horny flies.
So maybe you can talk a little bit about how it is that something like a fly lusting after its mate is related to these internal states. Why is that such an interesting way to frame the problem? Like, if you're saying, I have this problem, I want to study internal states and I want to understand how they work, why is the fly problem with pheromones and all the rest of it a good system for studying that?
Yeah, I think it comes down to two things. The primary one is that it's actually one of the most overt and noticeable internal states, I think, that I have observed in nature.
Right.
So what happens, right, is that a male fly walks around the world. He lives his life. He spends it mostly on fruit. He sometimes flies vast distances, but in general, he congregates with other flies.
Right.
And he sees other flies all the time. And they're not particularly interesting if he's not in the right mood.
Right.
But then if you just watch flies, even at home, right, sitting on an orange or something, you'll notice that sometimes the male's behavior suddenly switches. And all of a sudden, he starts following another fly. And this is a female fly. And this means that he's recognized her as a female and become sexually aroused. And when he gets into this horny state, all he cares about in the world is really this female. He will follow her for 10, 20, 30 minutes until she either leaves or they copulate. So somehow the visual profile of just this female fly takes on a completely new meaning to the male, and she becomes all he cares about.
Now that's interesting. The visual profile changes. When you say that, what are you driving at? Is it that suddenly this female that they might have seen, the male might have seen over and over and over, changes in some sense what the female means to him? As in there's this switch flips. How does that switch flip?
Yeah, so it has to flip inside of him. Nothing is flipping outside, right? The female profile looks the same. It's like you're walking down the street and if you were just hanging out, you're going to dinner with a friend, someone walking by on the street doesn't bother you. You don't pay attention to it.
Right.
But if you are, with your girlfriend or boyfriend at home, and all of a sudden, you similarly become sexually aroused. That also takes on a completely new meaning to you.
Right.
And that becomes, an object of focus.
Right.
And that's the exact same thing in flies.
Got it. So now, like what one of the interesting things is that it's so reliable at an experimental level. You need to have a switch you can flip reliably.
It needs to happen, if you're going to study this event 10, 20,000 times, it has to happen every time.
Right.
And the more reliable it is, the easier it is to study and to, pin down what is happening inside of the brain. Because you also know that if it's not happening, then something's wrong.
Right.
Something's changed. So if you, mess with the fly, or even with the fly's brain, and you notice that he's not doing this anymore, then you can very reliably say that this is because I changed something. It's
Right.
not just because it didn't happen this time. I flipped a die and it didn't happen.
Absolutely.
It's much easier to work with the statistics and to, reliably build up.
So I'm wondering, you have a chemical switch that you use though when you're in lab? Before the we can get into the fancy optogenetic stuff, but first you have a chemical switch that's available to you. Is that correct?
Right. So the male fly typically he tastes the female. So flies are cool because they taste with their legs.
Wait, what?
Yeah. So flies have all their gustatory receptors on their legs.
Are they like similar to ours? In how similar are they to our taste buds?
They're quite similar. Yeah, absolutely. They are chemical receptors that are recognizing, mostly hydrophobic things.
And how do we differentiate between taste and smell?
So smell is essentially just aerosolized particles, right? Mm-hmm. So these receptors aren't covered by mucous membrane. They don't pick up on, volatile chemicals that are just floating around in the air. They have to come into direct physical contact on a surface, essentially, with the object in question.
So this is key, right? That smell and taste are both chemical sensors, but I guess what you're saying is that smell is specifically for aerosolized chemicals,
exactly,
which are smaller. Like, what defines an aerosol, like an aromatic aerosolized chemical?
I actually don't know if aerosolized was the right word, but volatile at least.
Volatile, right?
Yeah.
They're small. They have rings often. They have carbon rings.
They often do. And they just have low boiling points, right?
Right.
They essentially evaporate into the air, just like water does if you boil it.
Got it.
Yeah.
And so, and the taste is for things that don't do this. So what you're saying is that the pheromones on the female do not waft or go off in the way.
They're not. They're big, heavy, fatty chains.
So how does a male fly get a taste?
So he walks up to a female and he just puts his foreleg on her belly. And he does this all the time. If you watch flies, that's all they do. They walk around, go up to one fly, put their legs on them and say, okay, is this a female? And there's two key things he needs to recognize, right? One is this a male or a female?
Right.
If it's a male, he's not interested. Males actually have another component on them that is actively aversive to the fly and tells him to go away.
Interesting.
Females of the same species have pheromones that are attractive to the male. Females of different species have pheromones that are aversive to the male. And that's really important because a male is about to spend 20, 30 minutes of his relatively short life trying to convince this fly to copulate with him. Right. If she's not of the right species, that is a wasted 30 minutes.
Right.
So it's actually a huge metabolic investment for the male fly to do this. So he has to get it right the first time.
You're doing a sense originally to say, is this worth investing in? Because if not, I'm about to waste tons of time and energy.
Exactly. And maybe after we've talked a little bit more about, how we think that this process actually happened and how this fly's courtship happens, we can come back to the pheromones of other flies that are not the right one. Okay. Because I think there's some really cool implications of how this is occurring.
Great. Okay, yeah. I'm excited about that. So we'll stick a pin in that.
Yes.
So now, but this has some cascade that switches an internal state. So how do we get from a pheromone, a chemical that tastes on their tongue arms, and then to a change in the brain state for the fly?
Yeah. So over the past I guess there's two aspects to this answer. One aspect is that, maybe 20 years ago, it was discovered that almost all of the neurons in the fly that are specific to one sex. So courtship is only performed by males. Females don't do it. So we think that something in the male brain is different from the female brain.
Mm-hmm.
And it turns out that most of the neurons involved in male-specific behaviors are marked by expression of a specific isoform of a gene called fruitless, which is a funny gene because if you knock it out, males become fruitless. They can't copulate.
Got it. You fly folks always have very fun names for everything.
They're shaker. There's a bunch of them.
And there's some rivalry between the worm people and the fly people in this regard.
I think we have the better ones.
I have to agree. No offense to any worm people out there, but I think the fly people have much better gene names.
Yeah,
much more fun.
Exactly. Yeah, there's a lot of fun, like neurogenetics in flies that has happened. It's fun.
So it's a gene called fruitless.
So it's a gene called fruitless, and this is really advantageous if you're trying to study these neurons, right? Because you can tell which neurons are likely to be involved in male-specific behavior. And there's only about 2,000 neurons in the male brain that expresses fruitless, and we think that they actually form as a little subsystem within the bigger picture of the fly brain that consists of about 100,000 neurons.
Right.
So that's one answer, right? This makes it so much more tractable to study the male fly.
So not all neurons have or are expressing the gene fruitless. And because of this, you can actually narrow down the number of neurons that you're looking at to a reasonable number to study.
Exactly.
And you've done this.
So people have done this. And it's been super fruitful, no pun intended, because this gene is also involved in actually changing the way that neurons look in the brain, right? They change how it looks in the male brain versus the female brain.
As in their morphology, the way they're shaped?
Yes, the morphology of the neurons, which even the existence of some of the neurons. So because females, for example, don't have fruitless expressed in some neurons, they kill off these neurons during development. Whereas males don't. So you end up with there's a small part of the brain that is built differently in males and females in the fly. And that part is presumably the one that is actually responsible for these highly sex-specific behaviors that is only exhibited by the male or the female.
Right.
So that was one key element that people solved, right? Now we have a much more tractable system. We can work with 2,000 neurons instead of 100,000 neurons.
Great.
Yeah, super great.
Huge.
And then people before me, both in our lab and in other laboratories, have figured out that they've essentially traced the activity that happens from the sensing in the foreleg up until the brain. Where are these signals going in the brain? And it turns out that they are going to a population of neurons that only exist in the male, called P1 neurons. And P1 neurons are some of the weirdest neurons in all of neuroscience. There's 20 of them per side of the brain. As I said, they only exist in males. They have no known outputs. And presumably they do have outputs, but we haven't figured out any of them. If you do we have techniques for trying to trace from one neuron to another which neurons are this neuron talking to.
How is that done? I actually don't know. Can you?
So it's a complicated chemical way. So you have one neuron express essentially half of a receptor, and then you have every other neuron in the brain express the other half of that receptor. So only where those two receptors meet do you get labeling, essentially.
So every other neuron, you force one neuron to create half of a receptor, and another neuron, all the other neurons to create the other half of the receptor, and then only where those two neurons meet do you get a receptor, which then you can cause to fire by some special neurotransmitter that you design or something.
Yeah, so it's not a fully receptor in the neuroscientific way. It's not an ion channel or anything like that.
Okay.
It's like a cross-synaptic thing that links the two neurons together. So it's part of, the mechanisms that neurons use to actually hook up synapses.
Got it.
So these two neurons will link to each other, and that will cause a cascade of small molecules inside of the second neuron to activate, and that will go into the DNA and allow certain fluorescent markers that we've inserted into the DNA to be expressed.
So they will find each other. They will link together. That will cause a cascade of small molecules in the second, which then leads to the expression of a glowing protein and causes that neuron set to glow.
To glow, exactly. So all the neurons that are talking to this neuron are now glowing.
Great. And you can pick that up with your microscope.
With our microscope, exactly.
Beautiful.
But if you do this in P1 neurons, nothing.
What is this technique called, just for people at home to Google?
It's called the TransTango.
TransTango.
Yeah, it's another nice fly name.
Great Googleable term for people who are interested at home.
Exactly, by Gilliard Barnea Brown. So it was actually discovered relatively recently.
Wow.
In the past few years.
Very cool. So this is stuff that has only become possible very recently.
I think that almost everything we're about to talk about would not have been possible even 10 years ago.
Wow.
Which is really fun, but also it's crazy how fast neuroscience moves.
Right.
I think. So these P1 neurons seemingly not talking to anything, but they respond to
just for the recap real quick. Male touches the belly of a female, tastes it, and it sends a signal cascade up to a set of neurons, the P1 neurons, which are controlled in some way. Their morphology is controlled by this gene called fruitless that only males have. So only the males have the P1 neurons. And now what happens?
And now they are now responding to these pheromones on the female.
Great.
And the cool thing is that just like the behavior, males shouldn't court females that are of a different species. The pheromones of other species actually inhibit the P1 neurons.
Turn them off.
So same species, turn them on. Different species, turn them off.
Great.
And now the cool thing about P1 neurons is, which was only figured out in the past 5-10 years by optogenetics. And optogenetics, right, is that we express a light-sensitive ion channel in some population of neurons, and when we shine light on the fly, these neurons will start to fire.
Great. So you somehow in there. How do you get that gene in there? Is it through a virus?
It's not through a virus. It is through a vector. Yeah, exactly.
So it's
a piggyback gene that's essentially a small circular vector that's inserted into the larvae, and that will insert itself into the DNA of the fly.
So what, you take a little viral capsule?
Yeah, with transposons.
Got it. So let me see if I can get a short summary of it. You want to try your hand at a simple summary of this?
Essentially, you build a short little circle of DNA, and that DNA when it enters the cells will be cut up into now a string. If you cut a circle in one place, it becomes a string.
Yep.
You also cut in the DNA of the fly to have some open space, like a gap, and then that string is inserted into the gap.
And you use a bunch of viral mechanisms, because this is how viruses kind of do things, or how do you?
I'm actually not sure how we did the optogenetics how they were done in the first place.
Okay.
But yeah.
This is a new technique. I'll find someone else whose specialty
Someone
is this.
is better at this than I am.
Great. So we'll skip to the point where you have these genes.
Exactly. Yeah. So we have genes inside of here, right? They get expressed only in the right neurons.
Got it.
Because we insert them essentially under controls of random enhancers, and they will be expressed only in some neurons.
Got it.
So if we express them in P1 neurons and activate P1 neurons just with light now, a male fly will begin to court. And he will court anything that moves. He will court ladybugs, he will court spiders until they eat him. Everything else...
Who decided to do these things?
Yeah, everything else, they just make courtship the priority. And that kind of makes sense, right? If the pheromones are activating these neurons, the pheromones are activating courtship.
Got it.
And the other cool thing about these neurons is that you actually only need to activate them for a second. And males will do this for minutes at a time.
Oh.
which is again consistent with, I tasted the female, and all of a sudden I now have to continue to pursue her for a long time.
Even the absence.
Even in the absence of, I can't keep tasting her the entire time. He can't hold his foreleg on her belly the entire time. He has to stay motivated even when he can't do that.
Right.
So they have this long, interesting time scale that they promote this essentially state of sexual arousal.
Right.
And here I think we begin to get into affective seeming behaviors.
Right.
These neurons really cause the emergence of an internal state. And for the duration of this internal state, any moving object looks like a female fly to the male.
Right.
And he will lock onto her for the next 10 minutes.
Any moving object.
Essentially, yeah. Anything that is small-ish. It doesn't have to be super small.
Right.
I assume he wouldn't court like a moving house, but who knows?
So for instance, my understanding is that you all used just a black dot on a screen.
Exactly, yeah. So Drosophila courtship is a remarkable behavior. It's been studied for the past 50, 60 years. It's definitely the most studied behavior in the Drosophila behavioral repertoire. But one of the challenges was that it's really hard to look at the brain when a fly is running around.
Yeah.
So we needed some way to essentially allow us to look at these neurons while the fly is performing this courtship ritual, to understand how they are changing the way he's reacting to the world.
Got it.
So therefore, we constructed this virtual reality for male flies.
Male fly VR?
Male fly VR. And I really mean VR. He's running on a small kind of spherical, a floating ball, like a treadmill, but in 3D. When he turns right, the world shifts to the left to compensate for his motion. Really, it's the same coupling as a real fly would imagine.
Right. And it has screens all the way in front of it, all the way around his vision.
And the screens tiles his entire kind of visual field. And it took me maybe 6-7 months to build this.
Right.
And then with just the hope that if I activate P1 neurons here, will he court?
It's a crazy thing to build.
A stupid dot in this world. And he does.
You put a dot on the screen.
I put a dot on the screen, and she now moves autonomously. She moves like a real female running around the world. And the first time I put the male on there, activated P1 neurons, he runs after her in this 3-dimensional world for 30 minutes.
Her being the dot.
Her being a dot. if just a black sphere. And the first experiment I think we tried, he chased her for 16 meters on this ball, which is about equivalent of a human chasing another human for 28 kilometers over half an hour. Just to tell you how crazy he is about her. He is fully convinced that this is the one thing that matters in his life.
Right. Just bolting.
Yes, like nonstop.
That's hilarious.
Absolutely bizarre.
And this is so if I'm imagining this correctly, you have a fly on a little ball, you have screens all around him, you have a black dot on the screen, and you flash a light on it. You flash a light on the fly's head to make it horny.
Yeah.
And then it chases the dot around the screen for the equivalent of 26 kilometers over the course of 30 minutes. Yes. If you compare body size to distance traveled.
Exactly. Absolutely bizarre. But now we had, right? It's all of a sudden a much more tractable problem, right?
Now it's still, so you can look at its neurons.
Now it's still. It's standing on a ball. I can now open the fly's head and look straight into the brain. And that was essentially our goal, right? We were trying to understand how do P1 neurons change this dot from just a random moving sphere to a target of desire.
Mm.
And what really facilitated that kind of our investigations into that is really 2-photon calcium imaging, right? Which is the technique that allows us to look at many neurons while a male is standing still.
Right.
And there's something that we express in these neurons that is sensitive to the amount of calcium in them.
Got it.
And neurons, when they're active, have an influx of calcium. So we can use this as a kind of proxy for neural activity.
When it glows, it's firing.
When it glows, it's firing. Exactly. And so the first place we started was actually trying to understand just how is visual information changed, right? And maybe it's kind of a stupid place to start, because why would your representation of the world change? That's not what you would expect, right?
You wouldn't expect that being horny changes how your eyes work.
Exactly. Unless you're Plato. But
why would Plato believe this?
No, I'm just thinking about, the world is not as it appears to us.
Right. Oh, yes, the Plato's cave version.
Yes, exactly. So we looked at visual neurons, actually, a population of visual neurons that were recently described to be important for courtship. So if you silence these neurons, if you shut them down, the male fly has trouble following the female.
Mm.
And surprisingly, when we looked at these neurons, before the male was horny, they didn't respond to the outside world, essentially at all. Some smaller responses, but essentially they are just quiet. Then as soon as you activate P1 neurons to put males into the state of sexual arousal, these neurons start to respond every time the visual target moves on that side of his essentially field of view. So if it moves over the right eye, the neurons in the right eye will respond. And this, coming back, right, kind of surprising, because it means that the fly's perception of the outside world, not just what he chooses to do with information coming from the outside world, but his very perception of the outside world depends on his state of arousal.
Right.
I think that's, it's kind of counterintuitive. It's surprising.
That is really counterintuitive, right? The P1 neurons go and change the behavior of some visual neurons to actually change the way that the visual signal of a female fly or a black dot on a screen cascades down to the motor neurons. How direct is this connection between the eyes and the motor and the walking? Yeah,
so it's actually cool. So if you activate the visual neurons on only one side of the brain, the male turns in that direction.
This is kind of now we're getting closer to what precisely you have started to submit a manuscript for, or doing revisions for right now, for a paper potentially, hopefully coming out in Nature. We don't know yet, but fingers crossed.
Yes, exactly. Always fingers crossed. Jinx it. But exactly. So, I mean, the first big discovery, right, was that the fly's perception of the world changes.
Right.
And if you activate those neurons, the male turns in that direction.
Yep.
If you silence those neurons, the male can't turn in that direction.
Oh.
So these neurons really seem to be the effectors that drives a male to turn left and right to allow him to follow a female.
Right.
Which is kind of surprising. So if you actually trace these neurons, at least in the female, where we now have a connectome.
What's a connectome for everyone at home?
Sorry, a connectome is essentially a map of every neuronal connection in the brain.
Right.
So the fly brain has about 100,000 neurons. We have about 60,000 of those now mapped. Who's talking to who? Which is a crazy achievement. I mean, of just vast scale. It's insane that it was actually finished almost.
It's rather shocking.
Yeah, absolutely. It's hundreds of terabytes of data. Nightmare, but very cool. And at least in the female, we can see that these neurons also exist, these visual neurons. And they're only two synapses. They only have one neuron in between them and motor neurons that go into the legs. So these neurons are really tightly coupled.
Oh, there's only one neuron in between eye and leg.
I guess there's two. There's only one neuron in between eye and leg, the neurons that go into the leg to tell the neurons in the leg to move.
Got it. There's two. But the motor neurons are just the neurons that are coming up with kind of calculating the precise way to move the leg, which then tell the neurons in the muscles to actually flex.
So exactly.
But that's not very far.
No, it's very close.
Is it close do you know, is it that close in humans?
Not known. Maybe there is something like that in a human. I don't think in general
I suspect not.
Probably not.
But
and this is also not the case with most fly visual neurons.
Oh,
this is probably a kind of unique property, or at least these neurons likely have a privileged, position in the visual hierarchy.
These neurons get to, in some sense, if I'm interpreting this correctly, are these neurons kind of overriding the rest of everything else and saying, no, we're very important visual neurons. This is a priority. We have to chase.
Yeah, we think that's actually the case. So even flies naturally have this behavior, right? They correct for, because they're flying often and there's wind. If a fly is blown to the left, the world, right, will shift in front of him to the right. And then flies have this evolved kind of natural response. They turn backwards against that motion to kind of correct their flight path. If if you're blown off course, you got to go back on course.
Right.
These neurons suppress that even. The most primitive response from the fly brain to just, the world is straight is suppressed by these neurons.
You're no longer orienting around the world. You're orienting around the female you're
chasing. The female is all that matters to the male when he's sexually aroused. So we think that these neurons really take over everything. And we don't know what happens to the rest of the fly brain, actually. But it's a cool idea that most of it is probably shunted.
Right.
Which is bizarre.
That is super bizarre.
Yeah.
Do these flies do song?
Yeah, so they sing towards this dot. So flies have this, when they're a part of their courtship ritual is extending one wing and vibrating it them to sing a species-specific song. And these flies will sing to this fictional dot. But we don't think it's coming from these neurons, actually.
Right.
Because if you silence these neurons, the male will continue to sing, but he will not track.
So that's notable, right?
Yeah.
Because it means that it's still in a given internal state, namely the courting or the horny internal state.
Exactly.
But that's why, is that why you use the word can't turn right?
Exactly. Yeah. So we think that essentially the male's internal state is essentially setting up the behavior before it's happened, right? The male's brain is poised to court this fly before he's even seen that fly. And now whatever moves in front of him, he will execute that behavior.
Right.
So that's why I say that he can turn left. The implementation, the kind of motivation to do so is likely still present.
Right.
But it can't be executed because these neurons can't couple to the motor neurons.
Because you've turned them off.
Because we turn them off.
Wow. How do you turn them off?
Similar to how we activate neurons. It's essentially just the inverse.
Is the inverse.
Yes, exactly. Also with light.
Got it.
Yeah.
Oh man, that's absolutely wild. So then you promised me that we would talk about other flies and other rituals. Are we there yet, or is there more to?
I guess there's two more kind of cool things, which is that P1 neurons actually seem to carry this representation all the time. They're not just activated by pheromones and then turn off. Continuously, they fluctuate up and down, and their fluctuations predict exactly how strongly the visual neurons are going to respond. So we think that they are essentially acting like a rheostat, right? Tuning precisely how strongly these visual neurons should respond when they will see motion soon. So we think that P1 are essentially carry an ongoing representation of the male's courtship drive.
And what would be adaptive about always having that up, like having that kind of tune throughout the day?
Yeah.
What's the reason in nature you would want that?
Because we think that this system sets it up, right? If there's an ongoing, so P1Rs are mostly off.
Right.
They're only activated in the first place by the female pheromone, and then they kind of oscillate up and down for a while, predicting. And the cool thing about setting up a system that's that, instantaneously sensitive, it's if something depends on something being on, that something can also be shut off. And that's a more flexible way to, change how you respond to the world than to just say for the next 30 minutes, these neurons are on. And what's advantageous is because I told you that other females actually inhibit the P1 neurons. So if a male were to walk around the world courting first a female of his own species, and then he walks up to a female that is not of his own species, that will quickly inhibit the P1 neurons. And because the P1 neurons are inhibited, it will turn off the gain of these visual neurons, making the male blind to that female.
Oh.
By extension, right? If you can continuously tune the kind of sensitivity of these visual neurons, you can make the male essentially only see females of his own species. This is kind of clever and primitive way to set up a system to just blind the male to inappropriate females.
As in, once it gets close enough and it samples, it says, "Oh wait, this is the."
Yeah. And males
wrong.
do this all the time. They run around and taste different flies.
Right.
And we think that is precisely because these neurons are easily excited, but they're also quickly shut off.
So the fact of the matter is that they're actually always fluctuating up and down to get it to initiate with something out there, and then figure out taste. Okay, no, this is it. So this isn't it, or this
keep going. Yeah.
Or keep going.
Yeah.
So
exactly.
it has a baseline amount of activation in order to. Is that right? Does that have a baseline activation, or
so it typically has no activation at all. But once the male has recognized that there is at least one sensible female, one female of the same species around, then they will have a baseline level of activation.
Oh, meaning there are females in the vicinity.
So I should be sensitive to that. I should start investigating and start kind of chasing after different ones and taste them.
Right.
Yeah.
Right. Because they're in the vicinity, now I'm gonna start looking.
Exactly.
Like it's like I'm at you suddenly have I'm at a singles bar. Good
exactly.
to know.
Exactly. You shouldn't do that if you're at work.
Right.
Yeah.
But once you realize context,
in
I'm
the
at
right
a singles bar.
Yeah.
You're in the right environment. This is the place to go.
Exactly.
Right.
Yeah. So it's funny. It's bizarre.
No, it really is, and it does kind of get you. You're right to say that it's a real, or maybe this is maybe I'm putting words in your mouth, but at least as far as I'm looking at it, it's nice to see the very simple version of what must be much more complicated patterns happening in more complicated animals. So getting to like the very simple version that you can of course compound endless complexity on top of, but getting down to just like, okay, how is it that this happens in a fly? It gives you some information as to what types of things you might look for higher up, with more complexity added higher up the tree.
Absolutely, I think that's completely right.
So, if you had to guess, are there any animals that are good follow-ups from Drosophila? So once the imagine you have the ultimate success, and now everything about fly courtship is figured out, which I know we're a long way from there, or maybe not. I'm not sure. You'll have to comment. But what would be the next species that you'd be interested in? What would be something that you would think would be similar enough that it would be relevant, but different enough that it would be interesting to study? Open-ended question, and no wrong answers, obviously.
It's a good question. I guess how much are we weighing tractability?
Right.
Some of the most, I think these kind of internal states are particularly exciting in social animals, because there's so much richness in social interactions. So even, a small step up is the honeybee, right? Extremely like eusocial animals, with relatively similar nervous systems to the fly. Then honeybees are very difficult to study, just very untractable. But
because they live in
hives, they depend on each other for survival. It's not super easy, but I think we'll get there.
Right.
But these kind of, it's easy to think, this is, of course, it's that simple because it's a fly. But the reality is that even much higher order animals have these kind of behaviors. So one of the classics, right, is Konrad Lorenz, who's kind of notorious ethologist, won the Nobel Prize with Nico Tinbergen in the maybe 60s, I think.
What's an ethologist?
Ethologist is someone who just studies the natural behaviors of animals.
They go out and they
go out and watch.
So for the audience here, we had Eric Ramos on, who's an ethologist. It would be classified as an ethologist. He goes out and watches dolphin behavior. That would be an ethologist?
That would probably be an ethologist, exactly.
Great. Okay, cool.
Exactly, the study of animal behavior. But then I think it has some weird implications of exactly your kind of frame of mind as well.
Got it.
But anyhow.
I'll go ask Eric later today.
Yeah, if he classifies himself.
If he considers himself this way.
But I think we drew so much inspiration from these ethologists and their writings in the 50s and 60s. And for example, there's Konrad Lorenz spent years watching geese in the wild. And when a goose is nesting, and an egg rolls out of her nest, she will pull it back. But she's actually remarkably insensitive to what she's pulling back. If you roll a billiard ball in front of her, if you roll a basketball in front of her, she will think that's an egg and pull it back into her nest. And I think there's a lot of these examples where animals are kind of just stupid, right? Their brains are so hardwired for these innate behaviors that this is the most important thing that I need to do for, survival, essentially. That it overrides everything else.
I think the nightingale exploits something pretty similar. Do you know about this?
No.
I think it's the nightingale. I could be getting the species wrong, but they go in and they'll lay their egg in the basket of another species of bird.
Amazing.
And as soon as they hatch, first of all, the first thing, they hatch early, so they're pre-incubated somewhat. So they hatch early, and as soon as they hatch, their first thing is they push all the other eggs out of the nest. But it's a hack because the bird species that they do this to, they're so attuned to the red part of the inside of the mouth of their young that when the nightingale baby opens its mouth, they'll start feeding the nightingale baby.
Oh my God.
And they'll do this for as long as the nightingale stays in the nest.
Yes.
And it will stay there until it is far larger than the parents. So you have this insane situation where this thing that murdered the children of this one other animal is sitting in their nest, bigger than the parents who are going and hunting for food and bringing it back for the baby.
And same with, there's these moths, right, in Eastern Europe that have themselves carried into the to ant stacks. And the ants will take care of them throughout their development and feed them over their own young. And, there's numerous examples of animals just exploiting the fact that animal behavior is really hardwired when it comes down to, especially the other four Fs, right?
Right.
Fighting, fleeing, foraging, and fucking.
Oh, is that the... I wasn't aware of these four Fs.
That is the real four Fs.
I didn't know I had stole this from somewhere.
I thought it was a clever ploy on your part.
I didn't even know it was a ploy.
Amazing.
Oh, very fun. So right, and so you have these. It seems as if a lot of animals, if they can discover, like in video games, you have sometimes people will discover an exploit.
Yeah.
And they'll use that exploit to consistently win. And then eventually the mods will come in with a software patch to fix the game because they don't want that exploit.
No,
but there are no mods in nature. And so these exploits just get discovered and then used by other animals who have discovered an exploit, which is so fun.
There's some balance, right? Like if it were to work too well, then, that species would have to evolve a way to get rid of it. But if it works, relatively to the advantage of both, that's fine.
Right. I always joke that Darwinian truths are truths that are true enough.
Yes, exactly. That is very good.
Or they're true insofar as they're useful.
Yes, absolutely.
It's not the game of. It's so funny. I used to work in and around the Silicon Valley, and you would hear sometimes. An young computer scientist or a biologically enthused computer scientist would say something like, "Oh, well, evolution's the ultimate optimization algorithm. It's the best way to..."
Yeah, he's definitely not.
I would say, no. You computer scientists are way smarter than nature.
Good enough works, right?
Good enough works,
Yeah.
and it's good enough over large time scales.
Yes, absolutely.
But these, and then there's also the imprinting, right? Is imprinting similar to this?
I assume that it is in some ways. I think.
What can you describe imprinting real quick?
Well, imprinting, right, is it's especially observed in birds. I think
it's in ducks.
In ducks is
probably a lot of different birds. I'm sure.
Where when they hatch, they become very attached to the first, essentially. I don't know what it's driven by, if it's motion or warmth, but essentially the first animal, or I think in some cases it can even be objects that they see after they've hatched, they become very attached to and will follow around for their young adulthood.
Right.
And, you've seen ducks out in the streets. They follow their mother on a kind of line. And if ducks are, if they hatch without a mother and next to a human or something instead, they will imprint on the human and follow the human around instead. So actually, if you at a lot of bird care facilities where they help damaged birds or birds with broken wings or something, or rescue eggs, the caretakers have to wear big masks, black masks over their faces to avoid the hatchlings from imprinting on the caretakers.
Oh,
yeah.
I didn't realize that. So they have some sort of maybe not even primitive. How do they see a human face as a face?
That's don't know.
Really interesting,
but
right?
It seems to be then kind of face oriented.
Something about two eyes.
Yeah, maybe.
2 eyes is enough.
Yeah.
Because we don't have beaks or anything like that. Our faces, I would think that we're so different. But
no, exactly. And it's clearly not just motion because, they're still moving. They just don't have faces.
Right. So there's something about the 2 eyes or something. That would be interesting to try to figure out what precise features on the face or what it is.
Yeah, absolutely. It's a good... I'm surprised. Someone must have done it.
Someone has to. Yeah. Maybe worth Googling afterwards.
Yes, I think so too.
Fascinating.
But I think, we also see these... Sorry. We see these things in, we can find similar themes even in our own daily lives, right? When you are angry, you also become irritable.
Right.
And things that typically don't piss you off start to piss you off.
Right.
And it's almost like your brain is generalizing behavior too far.
Mm.
And I think that especially when it comes down to these innate behavioral programs, that is really common.
Mm.
And those innate programs are often associated with, as you have brought up, affective states.
Right.
And there, I think the link lies between, these affective perceptual states in humans and these primitive states in the fruit fly.
Mm.
To me, they are probably building blocks of the same phenomenon.
Right. I tend to agree. And in fact, I think the thing that's interesting, and actually I would argue, and this is my claim, is or I'm really stealing this claim out of some of the things I'm reading out of Jock Panksepp's Affective Neuroscience book. It's a really cool textbook, by the way.
Yeah.
Highly recommend it. I like it a lot. But it's a bit old now. I know biologists. Here's a difference between biology and math is we have textbook culture where we love our textbooks. And because the informational half-life for math is for almost forever.
Yeah.
Something that was shown 500 years ago is still just as good now. And in biology, the informational half-life is closer to 20 years or something.
Tops.
It tops. Every 20 years, half the things you thought you knew were wrong. And now we have to relearn and re-understand.
Yeah, absolutely. But I think that's what's so... I would say that I've been very inspired by a lot of classic biology. And
right.
It's something I like a lot. But I think particularly these ethologists are notable because their observations of nature are always true.
Mm. There really isn't much of an informational half-life on just observing.
No, if anything, you can turn to them a lot for inspiration and to find, descriptions of curious behaviors that imply weird forms of information processing, I think.
Right.
And that to me is really fun and it's inspiring.
That's a great point.
It's not dying. It's still true that geese do this.
Right. That's a great point. I never thought of the fact that if you were to take the mechanisms out of it and you were just to look at the ethology, that the information half-life of ethology is very similar to that of like physics, say, which is quite long.
No, absolutely.
Another thing to note is that physics didn't always have a long informational half-life.
I didn't know that.
For a long time.
Yes.
Right. For a long time, people were making mistakes more, and it was the informational half-life got longer as physics progressed as a field. It was a young field for a while, and I think this is actually something that's really, really interesting. Is these days physicists talk, theory comes first, experiment comes afterwards. But if you look at early physics, it wasn't that way. Early physics used to be where the experimentalist would see something first, or they would make an observation in nature first, like an ethologist, and then they would try to figure out what's going on. So it's actually a very recent phenomenon that the theory comes first in physics.
Do you think that is because, do you think that fields go in waves? That there are periods of quick change and then periods of slow? It feels like, in the 1800s, someone said, like, we're done. We have nothing left to discover in biology.
What a crazy...
So, yeah, it's a crazy concept, but maybe that's why. Do you think that...
I certainly think that there's some punctuated equilibrium happening here.
And biology seems to be at the midst of its most, heavy flux right now.
There's a reason I switched fields.
Yeah.
Really?
Yeah, no, they understand. That's...
I just could read the cards, and I said physics is other. There's some realms of physics. I'm not saying all physics has stopped, but physics has slowed, and I'm seeing biology ramp up.
Yeah.
It's really massive, very quick changes are happening. And that was exciting to me. So I switched fields for that reason.
No, absolutely.
I suspect that what we'll see actually is a broadening of the informational half-life of biology.
Yeah. I mean, that's the hope.
I should define the term. Gosh, I'm being a bad host. I'm not defining the term. The informational half-life term is just how long it takes for half the information that you have accrued to be not irrelevant, but old news. It's no longer quite accurate. It's no longer the way that we think about things.
Right.
And of course, anyways, so I expect to see a broadening of the informational half-life of biology. But it's interesting to think that ethology doesn't really have nearly as short of one.
At least maybe longer, it seems.
Yeah.
Maybe not conceptually, but at least, observed. It's an observable truth in a sense, I think.
Absolutely.
An apple still falls, right? That's not going to change.
Same
in physics. Yeah.
Same in physics. It's an observation is forever. The other thing is I think one of the reasons that drives theory to come before experiment in some ways is the cost of the experiment.
Yes, exactly. Physics is in a state where, big revolutionizing experiments take a lot of money, I presume.
Right. Tons of money.
Yeah.
Like government can only fund one project every 10, 20 years level of money.
Yes.
So where biology, the amazing thing is you can still make big discoveries in a lab that is just you and a few other people.
Yeah, absolutely. No, it's a big difference. And then we are overprivileged in having, probably too much funding, but even that is like a joke amount compared to physics.
Right. Yeah. It really is. And that's the strange kind of interplay between, I think, age of a field and the informational half-life of that field.
Yeah, that's true. It's also something I find it very fun to, how far can you get with little money? I think you can make a lot of biological observations and discoveries with, on $10,000, I think you can go quite far.
I think you can do quite a lot with and this is what I've been playing with, is just a computer terminal and the data that exists in the world already.
That's absolutely true, yes.
There's so much that can just be done off of the data that exists.
That's true.
Because in some ways we have a wealth of data. Maybe this is something you could comment on, is that there's like this massive wealth of data. There's, with omics data, all the genomics and connectomics and the rest of it. And it seems as if it's almost more data than you could ever use.
Yeah.
I mean, what do you think the path forward on utilizing any of this data is? I know this is I'm asking an extraordinarily broad question, but
no,
with the connectomics data, how is that going to get used?
No, I think it's a huge question, and I think there are too few constraints in how we approach that data right now. We don't know enough quite about biological systems to infer function from structure. Just because you know which neuron is talking to who doesn't mean you can fully understand the flow of information.
Right.
In the worm, for example, right, they have 209 maybe neurons or something. And the connections between every neuron in the worm brain has been known for 40 years. We still have no more of an understanding.
Here's a good question for you. If we know everything about the connectome, do you think that's enough information?
No, absolutely not.
What else do you need? I don't really believe what I'm saying here, but I'll play this. I'll play this character for you. But if we know every connection between every neuron in the brain, why? That seems like it would be. It seems like it would be enough, doesn't it?
Because neurons can talk to each other in many different ways.
Ooh, tell me about that.
So, right, neurons have many different forms of synapses to begin with. There are electrically coupled neurons that don't even need a synapse. There are ionotropic synapses that depend on kind of ionotropic receptors in the postsynaptic neuron to quickly flow in ions to activate that neuron. But there are also a lot of G-protein coupled receptors that change properties inside of neurons on an ongoing basis. And that's even before we start talking about the neuron that is releasing the signal can release many, many different signals. Some neurons release classical neurotransmitters such as acetylcholine or the inhibitory neurotransmitter GABA, but there are also neuropeptides that can be released that go in and, change the properties of the neurons they're talking to. So one snapshot of the entire brain will never be enough, I think, to understand how brains work.
Right, because the connectome is just on average how everything's connected.
Yeah, at one time.
Yeah. And
it's a single frozen brain that's been sliced up.
And of course, you need to watch out those things as those maybe connections or everything else or the dynamics of that change in order to see these internal states that you're interested in.
I think so.
This is all very cool. One of my kind of wrap up questions is, who else is interesting in this field? Who else should I speak to, or maybe somewhat tangentially related to your work, maybe on campus or in New York City in general? Who would be an interesting person to talk to?
Oh, that's a good question. I had such a good answer for who you should talk to, but it wasn't in my field. So now I gotta.
Oh, we'll ask both questions.
Yeah, we'll start with the other one. The other person I think you should definitely talk to is Chung Liu in Gabby Maimon's lab.
Okay.
Who just had an amazing paper that I think you'd appreciate on, how the world is mapped into owl-centric coordinates inside of the brain of a fly.
Perfect. It's funny because I think you are the second person to recommend Chung. So I really have to reach out to this person.
Yes, I think you should. But the other person, I think talking with people in Daniel Cronar's lab is one super fascinating example of, these eusocial organisms that have much higher social pressures and much more sophisticated communication systems, of how they both, how these animals differ and how they have maybe different requirements for their kind of social behavioral states. But also, you can imagine that internal states don't only exist necessarily in individual animals, but are there essentially functional state that an entire ant colony could occupy, such that the colony as a whole reacts differently depending on their behavioral state.
Right. So sometimes you have to look at, with ants, the whole colony versus the individual, because they, as a group, have a certain group dynamics that are also important to preserve.
Exactly. That are almost richer than the behavioral dynamics of any individual animal.
Yeah, that is a tough job.
Yes, absolutely. They're doing a heroic job, getting the tools they need into the ants and just trying to make some amazing.
And then what's next for you? What you're finishing this paper, but am I asking too much to? I mean, maybe you're trying to keep things quiet and on the down low, but what is next for you in terms of things that you're about to dig into, or are digging into, or have data for, or kind of thinking about?
Yeah, I think there's so one thing we didn't really talk about that much was the fact that you can actually build a model of the fly's behavior.
Oh, actually, can we talk about that?
Yeah, sure. It's relatively quick. So you can build a model of the fly's behavior that simulates these neurons that drive courtship, and it almost perfectly explains what an animal does.
That's what's shocking about your paper when I read it.
It's like on a moment-by-moment basis of a fly just running around on a table. You can predict 10 minutes into the future what that fly is going to do. Every turn he's going to make, with the angle of those turns, his speed.
Because what you did, if I'm understanding correctly, is you modeled that neural system in a computer.
Yes, exactly.
And we're able to show that model matches right on to what's actually happening in the real world.
Yeah, what's cool is that we can even use these classical, what's called integrate and fire neurons that are actually, simulating spiking neurons. And even with that kind of excessively rich model, you actually produce the exact behavior that the fly will execute. So
it's a tour de force, man.
It's just weird that it's almost deterministic, right?
Right.
The fly is essentially operating like this model, which is crazy to me.
Now, one maybe challenging question is, you've seen these papers for the gut, the gut motions inside of a lobster. Have you seen
these? I have not, no.
Oh, well, I'll send you a paper at the end of this, which is very fun. They were able to show that many different models were able to show the same gut motions inside of a lobster. So how do you work out whether there's maybe infinite models that create these dynamics?
Absolutely.
How do you work out that the model that you have is really the model that's operating, not just a model that works?
Yeah, I think everything comes down to constraints, right?
Okay. And what types of constraints did you give yourself to guarantee or make it more clear that this was going to be the case?
I definitely don't think that we can ever guarantee that this is, the exact model. I'm sure there are a lot more added richness, or maybe there's even a different system. But some of the things we do is that we map, for example, how we measure how these neurons are sensitive to shifts in the outside world by directly recording from them. And then we build in those exact same sensitivities to the model.
Right. So your model is constrained by your observations.
Exactly.
That's huge.
And then it's also constrained by the functional connections that real neurons make in the brain are made in the model.
So you're only putting in connections between neurons that actually exist.
Exactly.
Well, that pretty much answers the question then, doesn't it?
It does. I mean, that's as far as we can go, I think. And then, it's really fun because we actually learned a lot from our model. It wasn't like, here is the behavior that a fly does, and here's a model that can replicate it. But we actually got a lot out of building that model.
Once you have a model, you now have a framework to think about next questions that could be asked.
Exactly. So one of the key things, for example, is that we noticed at first that our model was way too slow. Even with the right sensitivity to the outside world, the model was just reacting slower than the fly. And bizarrely, it turned out that if you actually make the model sensitive to the direction of motion, it was much faster.
Oh.
Because these neurons are sensitive, have a kind of long tail in what they're sensitive to, so the model became too slow. But if you make them sensitive to only one direction of motion, the model became much better. And then we went in and recorded from these neurons again and found that they are sensitive to the direction of motion and exclusively respond in one direction. And the animal's behavior is perfectly only driven by motion in one direction and not in the other direction.
This is one of these really fun things. When you have a tight loop between theory and
exactly,
experiment,
you can go back and forth.
You can say, oh, this theory predicts this. Okay, does that show up in the experiment? Great. Okay, we also saw this. Let's add that to the model. And that tight loop that can happen, the best science happens from tight loops like that.
Yeah.
That's
really fun.
Very fun.
Yeah.
And so now that you have this model.
Yeah, so now I think to answer your real question, I think there's two places I'm interested in going. One of them is that P1 neurons somehow maintain this for minutes to hours at a time. To build an active system that can maintain information for that long is really hard. Even theoretically, it's hard. So one question is, how is this information stored? How does a fly stay sexually aroused for minutes after he's realized that a female is around? I think that's a massive question.
Oh, that's the memory question.
It is
in some ways.
In a sense, but the problem is that here it has to be reversible.
It has to be an erasable memory.
Yes, exactly.
In some sense.
So
maybe memory is not the right word.
Maybe it is. It's probably a form of memory, but
or as a lot of things in neuroscience, probably any word.
Any word, exactly. Throw in anything. Yeah. But that question, I think, is very difficult, but very important for neuroscience, because I think if we can understand how these short-lived motivational states are built up, what they consist of, not just... I mean, this was very satisfying because we figured out how they actually change behavior, but what are they? What do they look like? And how are they constructed? I think is a massive question. It's also a very hard question, I think.
Right.
Because I think that more likely than not, it has a lot to do with subcellular mechanisms that are smaller than firing rates.
Smaller, like inside the neurons.
Of neurons. Molecules that can store information, that can change sensitivities in neurons for...
So you like this model of molecules storing information. I've heard people talking about this recently, and I was surprised that this paradigm was coming out. But
I think it's something that's been ignored almost for a long time in neuroscience.
But it seems almost offhandedly ignored. I'm not even sure if there's a good justification for ignoring the idea of like molecular storage of memory.
I totally agree with you. I think there's something to be said about the time wasn't right. Walk before you can run.
Right.
I think neuroscience, it was worthwhile to focus on spikes alone and that kind of communication, because it is true that it is likely how neurons communicate with each other.
Mm-hmm.
But I'm sure there is more richness to, ongoing dynamics within cells that help build up and help cells maintain information.
Wow, that's going to be interesting. Figuring out we know DNA is the genetic information storage, but what's the information store for the neurons molecularly is a really interesting question.
Yeah, exactly. For short periods of time, erasable things. It's fascinating.
What's the RAM in some sense?
Exactly. Yeah, absolutely. And yeah, if there is that, then the game changes, right?
It really does.
Yes.
I mean, that's a whole new field.
Yes, it really is. So I think that's a massive question that I'm super interested in. I think it's very difficult.
You reach for the stars, man. This is you have. I mean, you don't. I think you're in a good position to be asking questions like that. Luckily, this program gives us a lot of flexibility.
Yeah,
so
that's absolutely true. Yeah,
very cool. Generous. And you had a second question that you were interested in.
I guess the other thing is also, so flies actually, when they're in this natural environment, also run into males, and when they run into a male, they fight him instead. And fight him off.
I've also seen this in single bars.
Yes, no, flies on a mushroom. That's a single bar.
Yeah.
So to understand how,
what's you looking at, bro?
Yeah, no, seriously, they'll just kick the other fly in the face. It's really funny.
What is fly combat like?
It literally looks like fist fighting. With their forelegs, they're kind of slapping each other in the head. It's amazing. So, now we have one of these states, but how rich would it be if we had two? If we could study the interplay between aggression and sexual arousal, and how they kind of on a moment-by-moment basis decide whether a fly should court something or fight it. And do these systems exist in opposition to each other? Are they parallel systems that are just going simultaneously? I think that's also great.
I've heard people say that hate is closer to love than apathy.
No, absolutely. I think so. We just had a symposium called Love and Hate in the Fly. So,
oh, really?
Yes.
No way. So there it is.
So yeah.
That's an empirical question in some way. You can make that question empirical. You can say, how closely are these mechanisms related?
And one of the really fascinating things is that if you activate P1 neurons very, very slightly, flies become aggressive instead of begin to court.
Oh, so you might have a tri-state.
So there might be, maybe even the representation of aggression and sexual arousal occur in the same neuron, making them almost indistinguishable from each other, but somehow they yield different behavior. So trying to think more about the interplay between these is also a really interesting direction. So just recently we have made flies aggressive to virtual objects in this virtual reality.
Oh, just like
they tried to court other objects, we can now make flies by activating another population of neurons, make them try to fight this object.
Can you get them to fight spiders?
That's a good question. We should try that.
Get them to fight spiders or other types of flies?
No,
It could be good.
You can definitely make them fight
other flies.
Other flies, yeah.
Easy.
Yeah.
But
it's a good question. We should try this with other...
Instead of being scared. Because one question is, if I'm going to take it into my realm a little bit, because I work with human data, so I'm a lot more... I'm thinking about the human side of these things a lot. And, can you get just fear? Can you get anger to trump fear? Because anger and fear, at least in mammalian systems, seem to be inhibitory.
From each other. Yeah.
You don't choose to flight and fight don't happen simultaneously.
No, they are. Yeah, exactly.
You choose one or the other and you go for it.
Absolutely.
If these things, I'd be curious to know if this is the same somehow in the fly. And this actually was something that I was thinking about was that in some ways the cortex, and this is what the mammal system has that the arthropod system doesn't. Is it they're arthropods?
Yeah.
Don't really have, is the ability to have this like cortical breadboard which can switch and modify the affective states. So in some ways I suspect that the reason why affective states seem so much richer in humans or mammals in general is because they have the ability to rewire what those affective states mean to some degree by having this like more highly reprogrammable cortical layer on top of the midbrain.
Yes, I think that's probably true.
So
yeah,
I guess finally, is there anything else inside of your world that you want to talk about? Anything you want to promo? Anything that you're like? This is your moment to say anything that you want to say or soapbox on anything you want to soapbox about.
I would just say that maybe I would, if I took one thing away, I would say that maybe humans are more predictable than we think, right? Maybe brains in general are relatively predictable, and this kind of makes me hopeful that actually all behavior is explainable. It is not, maybe to summarize it, just free will is an illusion.
Well, with that,
cheers.
Cheers. Thank you very much for coming on today.
Thank you so much for having me. It's my pleasure.
Thank you for tuning in to this episode of Solutions. I hope you enjoyed listening to it as much as I enjoyed making it. Now, a few things that can help me out and help out the podcast in general, if you wouldn't mind, is please like, share, subscribe, and comment to the YouTube channel, and leave reviews on any of the podcast platforms that you may be listening to this on, including Spotify, Apple Music, etc. And also, please join our Discord. We have a Discord server where we hang out on Mondays during releases, or hopefully Mondays, we'll have to see. And we discuss the podcast episodes, and I hop on there and I engage with the community, and you can engage with other people who also enjoy listening to the podcast. So the Discord's a really cool place. I hope you guys can come out. I do math there every day at 7:00, so you're welcome to join for that. And that may be subject to change in the future if you're listening to this in the future, but for now, it is a wonderful place to come and hang out. So join the Discord, share, subscribe, like, comment on YouTube, review on the different platforms, and really, most importantly, share this with your friends, guys. If you like science, if you're enjoying the discourse, if you like hearing two scientists talk to each other on a podcast, give me some love and share it with your friends, and that will really help us grow as a channel and help me keep producing this content. So I thank you, and have a wonderful rest of your day, night, or evening, whichever time it may be. Signing off, Mason Hargrave.